Welcome to the Deep Dive, where we unearth those surprising nuggets of knowledge that help you truly understand what's going on, even in the most overlooked corners of the natural world. Today we're plunging into a truly paradoxical group of organisms, the Heterobasidiomycetes. You might know them by their more whimsical name, jelly fungi. How can a group of fungi that often look like gelatinous blobs be both a farmer's worst nightmare and the life support system for some of the world's most delicate plants? Our deep dive today, drawing heavily from Introduction to Fungi, 3rd edition, is all about unraveling that mystery.
It's a fantastic paradox, isn't it? While these fungi might seem, well, unassuming, they are incredibly diverse, and they play crucial, often surprising roles in everything from agriculture to medicine. Our mission today is to systematically peel back the layers of their unique characteristics, understand their intricate life cycles, delve into their varied ecological roles, and uncover their profound real-world significance. We aim to make even the dense scientific information clear and truly engaging for you, revealing just how much complexity lies within these seemingly simple forms.
Okay, let's unpack this. We're talking about jelly fungi, or Heterobasidiomycetes.
Yeah.
What exactly are these organisms, and what are the signature features that set them apart from the rest of the fungal kingdom?
Well, the terms Phragmobasidiomycetes and jelly fungi are indeed largely synonymous with the class Heterobasidiomycetes. What truly defines them boils down to, let's say, 4 key characteristics, each an intriguing adaptation. First, let's talk about their cellular connections. They possess what's called a complex dolipore septum.
Dolipore septum.
Right. Imagine it as a sophisticated, finely tuned gateway between cells, surrounded by a specialized structure called a parenthesome. This elaborate control point allows for specific substances to pass while regulating others, making it far more complex than, a simple opening you might find in some other fungal groups.
So it's not just a hole, it's an intelligent junction, kind of like a regulated doorway.
Yeah.
And their spore-bearing structures, their basidia, are also quite unique, right?
Precisely. That's their second defining feature. Unique basidia. Unlike the simple, club-shaped basidia of Homobasidiomycetes, which are the typical gilled mushrooms people usually picture, Heterobasidiomycetes have basidia that are often lobed and divided by internal walls, or septa.
Septa. Oh.
Yeah. These divisions can be transverse, oblique, or longitudinal.
Yeah.
These distinctive basidia are often called Heterobasidia or phragmobasidia, and they feature a very prominent spore-bearing stalk, the epibasidium.
And their appearance. That's where the jelly moniker comes in, literally.
Absolutely. Their third characteristic explains the name: simple, resilient fruit bodies. The architecture of their fruit bodies is often quite basic, without the elaborate caps or gills you see in many other fungi.
Simpler looking.
Exactly. But they have an amazing compensation. These simple fruit bodies are remarkably tough. They can dry out completely, becoming hard and brittle, and then when rehydrated by rain or dew, they swell up, become greatly gelatinous, and can produce fresh crops of spores.
Wow.
It's an ingenious built-in survival mechanism, allowing them to bounce back from desiccation.
That's a brilliant adaptation for survival in unpredictable environments. What about their spores once they are produced? Do they have any unique dispersal tricks?
Indeed, that's the fourth point, the secondary spore production. Basidiospores from most species have this fascinating ability to produce secondary spores.
Secondary spores, how does that work?
It means the initial spore, upon landing, can germinate to produce another ballistospore, which is actively discharged again, or passively released conidia, or even yeast cells.
So multiple chances to spread.
Exactly. It gives them multiple chances for dispersal and establishment, a truly versatile strategy.
This ecological versatility you mentioned seems crucial. Beyond their unique structures and spore dispersal, how do these jelly fungi truly fit into the broader ecological landscape?
Ecologically, they're quite the shapeshifters. Many Heterobasidiomycetes are primarily associated with wood and other decaying plant matter, acting as saprotrophs, which are crucial decomposers, recycling nutrients in ecosystems.
Recyclers.
Right. But a significant number also function as mycoparasites.
Mycoparasites. So parasites of other fungi.
That's right, meaning they specifically parasitize other fungi.
So some are literally making a living by feeding on other fungi. That's a niche.
It is. And it gets even more intriguing. Some species, particularly those within the Ceratobasidiales order, manage to walk this fascinating ecological tightrope. They can be necrotrophic pathogens of various plants, killing plant cells to obtain nutrients.
Okay, it's the bad guys.
Yet at the same time, they can also be crucial mycorrhizal associates of orchids.
Wait, partners with orchids?
Yes. It's a remarkable example of ecological flexibility being both a villain and a hero in different contexts.
Speaking of Ceratobasidiales, that brings us to the genus Rhizoctonia. This genus is a massive deal in the agricultural world. It's almost legendary for its dual nature. How can we recognize it, even at a microscopic level? And how do we classify something so versatile?
Rhizoctonia has some very characteristic hyphal branching that makes it quite recognizable under the microscope. Their branches typically arise at a right angle to the main hypha, and you'll often see a slight constriction at the branch point, with a septum, a dividing wall, located a little way into the branch itself.
A right-angle branch with a little pinch in a wall.
Okay, exactly. They also have those conspicuous dolipore septa we discussed. While these hypha are distinct, identifying the exact species can be tricky, based solely on morphology. This led to the development of the anastomosis groups system.
Anastomosis groups. That sounds like a sophisticated classification system. Can you explain how it works?
It's essentially a genetic compatibility test. Scientists pair isolates of Rhizoctonia with known tester strains in a lab. If the isolates are compatible, indicating they are closely related or genetically identical, their hyphae will fuse, or anastomose.
Fuse together.
Right. If they belong to different groups, they simply won't. It's like a genetic handshake to determine family connections, allowing us to classify them by their genetic compatibility rather than just their appearance.
Okay, that makes sense. So what does this all mean for us, especially for those of us who grow plants or care about our food supply? How does Rhizoctonia impact our lives, for good or for ill?
Well, the impact of Rhizoctonia on agriculture is unfortunately significant. One of the most important species is Rhizoctonia solani, which causes widespread soil-borne diseases. The classic example is damping off in seedlings.
Damping off, I've heard of that. Not good.
Not good at all. Imagine a young seedling just emerging from the soil, suddenly wilting, its stem near the soil line becoming water-soaked, losing its structural integrity, and then collapsing.
Right.
That's damping off, leading to the seedling's pre- or post-emergence death. On older plants, our solani can cause cankers and girdling on stems and root and foliar infections.
And it sounds like once it's in the soil, it's incredibly persistent. A true farmer's nightmare.
It truly is. The fungus can survive in the soil for several years as small, hardened masses of mycelium called sclerotia.
Sclerotia.
Yeah.
Like little fungal bunkers.
Kind of, yeah. These appear as black scurf on potato tubers, for example, making them difficult to remove. Another pathogenic species, Rhizoctonia cerealis, causes sharp eye spot in cereals, creating distinctive spindle-shaped lesions on stems and leaves.
So control must be difficult.
Because chemical control can be challenging for such persistent soil-borne pathogens, biological control shows a lot of promise. Researchers are using other beneficial organisms, including various bacteria and fungi like Trichoderma species, which can secrete powerful cell wall-degrading enzymes that effectively dismantle the pathogen.
Fighting fungi with other fungi.
Exactly. There's also the fascinating concept of soil suppressiveness, where certain soils naturally build up biocontrol organisms after continuous cropping, essentially developing their own immune system against the fungus. But what's truly fascinating here is how this seemingly destructive fungus flips roles in another critical area, orchid symbiosis.
Orchids, right? You mentioned that orchids are known for being incredibly particular, but Rhizoctonia, how does that work, especially if it's such a notorious plant pathogen in other contexts? It's like the fungus is both a villain and a hero.
It's an incredible partnership and one of the great paradoxes of the fungal kingdom. All members of the orchid family, some 17,500 species, rely on mycorrhizal fungi throughout their life cycle in nature.
All of them, wow.
Yep. What's unique about orchid mycorrhiza is that unlike most other mycorrhizal relationships, there's a net flow of sugars from the fungus to the plant, especially when the orchid seedling is establishing itself.
So the fungus feeds the orchid. Usually it's the other way around, or in exchange.
Exactly. Colorless, non-photosynthetic orchids actually rely on this external sugar supply throughout their entire lives. And yes, many of the fungi that orchids exploit are indeed serious plant pathogens like Rhizoctonia species. Some of the very Rhizoctonia strains isolated as pathogens of other plants can support orchid seed germination.
That's so counterintuitive.
Yeah.
So a tiny orchid seed, which looks like dust, completely relies on this fungus just to get started.
Exactly. Orchid seeds are incredibly tiny, lacking differentiated embryos or any food reserves. Without a soluble external carbohydrate source, they can only germinate to a limited extent, forming an intermediate stage called a protocorm.
A protocorm.
Yeah.
Like a pre-plant stage.
Sort of.
Yeah.
Further development only happens if a suitable soluble carbon source is added or if a mycorrhizal fungus like Rhizoctonia is present. Scientists can even demonstrate this with a clever split plate experiment. You can set up the fungus on one side of a barrier with a food source and the orchid seeds on the other. The fungus will grow across the barrier, still enabling the protocorm to grow, proving that carbon is being translocated from the fungus to the orchid, mainly as a sugar called trehalose.
Trehalose, okay. So it's actively piping sugar across. And how does the fungus physically interact with the orchid at a cellular level to achieve this transfer?
The fungus infects the orchid protocorm primarily through epidermal hairs. Once inside an orchid cell, the fungus's hyphae form a dense, coiled mass called a peloton.
A peloton, like in cycling.
Hey, yeah, sort of like a tight bunch. Initially, each fungal hypha is surrounded by the host's plasma membrane, known as the parafungal membrane. What's fascinating is the dynamic, almost unstable nature of this symbiosis. These pelotons are quickly degraded by the orchid, often within 24 hours.
The orchid digests them.
Pretty much, and the orchid cell can be repeatedly reinfected. This constant cycle of peloton formation and degradation is interpreted as a finely tuned defense reaction by the orchid against fungal invasion, ensuring the orchid benefits without being completely overrun.
So the orchid keeps the fungus in check.
The orchid, you could say, calls the shots, keeping the potentially pathogenic fungus in check while still extracting vital nutrients.
That's a truly sophisticated biological negotiation. Amazing. From that intricate dance with orchids, let's broaden our view. Beyond Rhizoctonia, there's a whole world of these jelly fungi with their own unique quirks. Let's talk about the Dacrymycetales. What's their most distinctive identifier?
The Dacrymycetales are primarily characterized by their forked or furcate basidia.
Forked basidia, like a Y shape.
Exactly. It's a very distinctive Y-shaped structure where the basidium splits into two prongs. Their fruit bodies are typically bright yellow or orange, often due to the presence of carotenoids, giving them a vibrant color, and they have that distinct gelatinous texture. They range from cushion-like forms, like Dacrymycetidales, to more upright, branched forms, like Calycera viscosa.
And how does their life cycle exemplify that resilience we talked about earlier, that drying out and coming back?
Well, take Dacrymycetidales, for instance. It produces two types of structures. You'll find soft, bright orange conidial pustules, which essentially break apart into numerous arthroconidia, easily dispersed by rain splash.
Spread by rain.
Right. Then there are firmer, paler basidial cushions that bear those characteristic forked basidia, each forming two haploid basidiospores. What's particularly interesting about their life cycle is how the parenthesome and dolipore complex, those cellular gateways we discussed, actually temporarily dissolve to facilitate the crucial passage of nuclei during decaryotization when two compatible hyphae fuse.
The gateway dissolves. Wow.
Yeah, it's an amazing example of cellular architecture adapting for genetic exchange.
That cellular flexibility is remarkable. Next up, the Auriculariales. These have a different kind of septation in their basidia, right?
Yes, the auriculariales are defined by having septate basidia, meaning their spore-bearing structures are divided by internal walls. These septa can be either transversely septate, like in the genus Auricularia, dividing the basidium crosswise,
Across?
or longitudinally septate, as seen in Exidia and Pseudohydnum, dividing it lengthwise. The latter are sometimes referred to as tremelloid basidia.
Got it. Let's focus on Auricularia, Auriculajudae. The Jew's ear fungus. That's a common one and seems to be a master of rehydration and spore dispersal.
It's quite distinctive. It forms rubbery, ear-shaped fruit bodies, often found on elder branches. Its fruit bodies have this remarkable ability to dry to a hard, brittle mass and then, upon re-wetting, quickly rehydrate and discharge spores within hours.
The ultimate comeback fungus.
You could say that. Its cylindrical basidia become divided into 4 cells by 3 transverse septa, each with a long cylindrical epibasidium extending to the surface, bearing a monokaryotic basidiospore. What's truly notable is that Auricularia's basidiospore at 20 micrometers or more is one of the largest propelled by the surface tension catapult mechanism for spore discharge.
So it really flings its spores.
It's a powerful disperser, and its spores have multiple ways to germinate. They can undergo repetitious germination, producing another ballistospore for a second chance at dispersal.
Another shot.
Or form minute lunate microconidia on small projections called denicles in nutrient-poor conditions. Or even direct germination by a germ tube on richer media. Very versatile.
If we connect this to the bigger picture, This isn't just a quirky fungus. It's one that has shaped human history in a unique way, isn't it?
Indeed. We're talking about Auricularia polytricha, often known as mu'ar, or cloud ear
Oh,
fungus.
dear.
This species holds the remarkable distinction of being the first cultivated mushroom for which we have historical records dating all the way back to AD 600 in China.
The first cultivated mushroom. Really?
That's what the records suggest. It's highly valued for its nutritional content and its unique chewy, rubbery texture, making it a popular ingredient in Far Eastern soups and stir-fries. Its cultivation continues to this day, traditionally on logs of broadleaf trees, or more recently in plastic bags filled with sawdust and rice bran. It's a testament to its enduring value and role in human culture.
Fascinating. The first documented cultivated mushroom. That's incredible foresight. Finally, let's explore the Tremellales, another intriguing order within the jelly fungi. What truly makes them stand out?
The Tremellales have a very defining characteristic. They possess a yeast-like haploid state in their life cycle.
A yeast state.
Yeah.
Like brewer's yeast.
Similar in that it's single-celled and buds, yes. Their basidia are divided by longitudinal septa, those tremelloid basidia we mentioned earlier. Also, their eukaryotic hyphae have a saculate parenthesome, meaning it's invaginated or pocketed towards the septal pore. They also employ a modified tetrapolar mating system, where specialized peptide hormones called tremerogens are exchanged between compatible yeast cells to trigger conjugation.
Hormones for mating.
Mm.
Complex stuff. What stands out to you about their ecological strategy, particularly how they interact with other fungi? Do they have a particularly aggressive approach?
Their ecological strategy is primarily as mycoparasites. You'll often find them growing on or very near the fruit bodies of other fungi or even on lichens, which they parasitize.
Okay, so back to parasitizing other fungi.
Right. Their mechanism of parasitism is quite unique, involving specialized hostorial branches. Now, despite the name, these aren't true hostorias that penetrate inside the host cell.
Not inside. How then?
Instead, these are modified hyphae with a swollen segment and thin filaments that contact a host hypha. They establish direct cytoplasmic contact by dissolving a small part of the host wall and fusing plasma membranes via micropore, essentially creating a direct bridge to siphon nutrients.
It's like tapping into the host's plumbing.
Exactly. Species of Tremella are well known for this, acting almost like fungal nutrient siphons.
So they're like fungal vampires, but with a highly specialized, almost surgical way of connecting. How does their complex life cycle unfold with this yeast-like state?
Their life cycle is quite intricate. Basidiospores can germinate by repetition, producing another ballistospore for broader dispersal, or they can form minute blastoconidia, which then bud to produce that characteristic haploid yeast state.
The yeast form again.
Right. When compatible yeast cells encounter each other, they fuse, reestablishing the dikaryotic mycelium, which then goes on to form new fruit bodies. This raises an important point. While some are parasites, others, like Tremella fusiformis, are highly valued for surprising reasons, moving far beyond their jelly appearance.
Ah, the silver ear fungus. Many people know this from Asian cuisine, particularly in desserts. But it's more than just a culinary ingredient, isn't it? What makes it so special?
Far more. Tremella fusiformis is another historically cultivated mushroom in China, cultivated for over 200 years. While it's certainly consumed as a dessert, it's also highly valued as a medicinal mushroom in the Far East.
Medicinal. How so?
It's attributed with properties like being life-prolonging, vitalizing, and even having anti-cancer effects. These benefits are thought to be primarily due to its ability to stimulate the immune system.
That's quite a claim for a jelly fungus. What's the biochemical basis for these effects, particularly what sets it apart from other medicinal fungi?
The attributed properties are thought to be due to the production of exopolysaccharides. Now, many medicinal fungi produce similar polysaccharides, but those typically have a 1-3 glucan backbone.
Okay, glucans. Common in fungi.
Right. In contrast, Tremella's unique exopolysaccharides have a 1-3 mannan backbone that's substituted with xylose and glucuronic acid. This makes them biochemically distinct.
Mannan, not glucan. Interesting.
What's quite intriguing is that these alloglucuronomannans actually have a structural similarity to the capsule of the human pathogen Cryptococcus neoformans.
Similar to a human pathogen. Whoa.
Yeah. So what this tiny structural difference reveals is how evolution can take similar biochemical building blocks and repurpose them, sometimes for medicine and sometimes, surprisingly, for pathogenicity. It's a striking example of nature's biochemical ingenuity and its potential for both good and ill, all within the fungal kingdom.
What an incredible journey through the world of the Heterobasidiomycetes.
Yeah.
From their unique jelly-like textures and the ingenious adaptations of their basidia, to their intricate life cycles and surprising roles as both plant pathogens and essential symbiotic partners for orchids.
Hmm.
Wow, these fungi are truly full of hidden complexity. They defy simple categorization.
Indeed. And whether they're breaking down wood, causing plant disease, nourishing orchids, or offering potential medicinal benefits, the jelly fungi remind us how adaptable and vital life forms can be, often in ways we're only just beginning to fully appreciate. The paradox of their simple appearance, hiding such profound ecological and biochemical roles, is what truly makes them a subject of endless fascination and ongoing discovery. There's still so much to learn.
Thank you for joining us on this deep dive. We hope you feel a little more well-informed and perhaps a lot more curious about the unseen world around us. Until next time.